The objective include brief introduction to various structures of prokaryotes, and also we'll talk about their functions. And in this part, we would talk about structures external to the cell wall only. Uh, we will discuss in a little more detail about the glycocalyx. This is an overall structure depiction of a bacterium where you could see that the cell has different structures. Some are, if we consider cell membrane as the most internal lining, the structures that are external to the cell lining include cell wall, capsule, then these hair-like structures, what we call fimbriae or pili, and then there's a long thread, it's called a flagellum. Uh, in this presentation, we would focus only on glycocalyx, and some part would be covered on the flagella. But before we talk about various structures, let's just go quickly through the functions of these structures. Plasma membrane, as you know, it provides a selective barrier. It is used for nutrient transport and also used in respiration and photosynthesis. Gas vacuole, there are certain bacteria that live in water, in seas, in ponds, and those that need light, they have evolved with these gas vacuoles, which keep them floating on a level that is required for the nutrition to be provided there. Next structure is ribosomes. These ribosomes are protein synthesis factories. Then inclusion bodies, they're also found in bacteria. These are storage of carbon, phosphate, and other substances that bacteria acquire from the environment and stores them and uses them on demand or on need. Nucleoid is the area where DNA is located. It's a genetic information. There's a space called periplasmic space. This space is basically external to the cell membrane. And there's a lot of enzymatic activity going on in this region. And it contains hydrolytic enzymes and binding proteins for nutrient processing and uptake. Next structure is cell wall. Its function is that because it is rigid chemically, it provides stability to the shape of the organism and also protection from osmotic stress. So, if a bacterium sits in an environment where osmotic pressure is more or less than the bacterial cell, cell wall protects it from getting damaged. Capsule and slime provides resistance to phagocytosis. Bacteria, when invade the body, cause the disease. They need to get out of the reach of the cells of the immune system. So, capsule and slime, basically capsule, helps these bacteria evade the immune system, go away from the immune system. So these cells cannot handle or cannot phagocytose or ingest or eat these bacteria and kill them. Fimbriae and pili, these are projections on the cells. They are used for attachment of this bacterium to surfaces and also transferring their genetic material, the DNA, from one bacterium to the other. This structure, what we call flagella, is used for locomotion. So, bacteria, if it has to move from one place to the other, it uses its flagella. It beats it by turning it either clockwise or anti-clockwise, and then it moves from one place to the other for seeking nutrients. Endospore is a special structure of the bacterium. It is formed under environmental conditions that are not very friendly for the bacterial growth. And this is a survival strategy by some bacteria. Not all of them are able to make these endospores. As I mentioned, that if we take cell wall, which is the middle layer here, as our landmark for studying structures that are external to the cell wall, and then there are structures that are internal to the cell wall. We see that there is a capsule, and then we see these fimbriae, or what we call hair-like, projections. And there are other structures also that include the capsule, as I mentioned, flagella, which is a long thread, not shown here in this picture. Flagella comes in two forms. Most bacteria have flagella, but spirochetes flagella is called axial filament. The purpose of the flagella, as I mentioned, is to make movement possible. Fimbriae and pili, these teeny fimbriae are these ones, hair-like projections, and pili could be one or could be two or could be many, could be up to 10. And these are used for DNA transfer from one bacterium to the other. Now, let's talk about the glycocalyx. It is viscous in consistency and gelatinous, gel-like. It surrounds the cell. So it is secreted by the cell and adheres to the outer part of the cell or the bacterium. It is composition-wise, it has polysaccharides and also proteins. So it's a combination of both polysaccharides and proteins, what we call polypeptides. And the composition of these polysaccharides and polypeptides vary from one organism to the other. And this variation, I will tell you, could be used for identification of bacterium. This glycocalyx is called a capsule when it is organized and is tightly attached, firmly attached to the cell. We call that glycocalyx as a capsule, and it has a role in the virulence. Virulence is the ability of the organism to cause damage when it infects the body. And this, as I mentioned, the capsule could also be used as identification of the bacterium. And if we take the capsule out of the cells and make a vaccine and inject into an animal, and animal would form antibodies against that capsule, which is polysaccharide and polypeptide. These are not normally present in the body, in human body or animal body that the bacteria infect. So body recognizes that as a foreign substance and induces immune response, making antibodies. So this capsule could be used as a vaccine. If the glycocalyx does not adhere to the bacterium firmly and is not very organized, we simply call it a slime. Now this is a bacterium here. You could see this is a negative stained slide that shows some of the bacteria. There's a bacterium. These blue or black cells here, they depict the organisms, and then there is a clear halo around the cell. This halo basically is the capsule. So if you want to see the capsule after staining, what we call negative staining, it is one of the techniques that we mentioned earlier. So a capsule could be visualized by negative staining. This is a colony, what we see on the nutrient agar. If we grow this slime secreting or capsular bacteria in the lab, the colonies look like these. See, kind of glistening and mucoid in morphology, in shape, and in appearance. These are some of the examples of not all organisms possess this capsule. Not all of them. Some do, and these are three examples that I selected. Bacillus anthracis it causes anthrax in animals, a very deadly disease, and it also can infect humans. Then there is another bacterium, what we call Streptococcus pneumoniae, which causes pneumonia in humans. Klebsiella is another organism not shown here. It's a rod-shaped gram-negative bacteria or bacterium that causes various mostly secondary infections in humans. Glycocalyx can also be used as a biofilm by the bacterium. So because it is a secretion, and then it's viscous in consistency, gel-like, so it would adhere to the surface. The bacterium, if it chooses to stay in one place where there's a lot of nutrition for the bacterium, so bacteria does not want to move. So it would secrete that glycocalyx as biofilm and then would adhere to the surface. Other than that, they also, when it is making biofilm, it also secretes extracellular polymeric substances, and they protect the cells. The cells make a colony, and all the cells are then protected by this biofilm. And it is very difficult to remove that bacterium if that makes a biofilm in the body after the infection. It's very difficult to treat such infections. So this glycocalyx can help the bacteria to attach to various surfaces for survival. This picture here shows a biofilm on the teeth, and people who don't brush their teeth very well, they end up with this biofilm, and then the bacteria colonizes their teeth and damage the teeth and makes those cavities and other tooth infections. This is a diagrammatic representation of a cell that secretes biofilm. So the cells attach to the surface, then it secretes these EPS, and then this EPS helps the bacteria colonize the surface, and the bacteria, they multiply in that area, making big colonies, and then they can discommate from these colonies. They can detach from these colonies, and then can go from this place to another place. And then again, this cycle could be started by, the first is attachment, then there's a development, this biofilm and colony making, and then, so this is the way these cells survive from one place to the other. Let's talk a little bit about the flagella, although we would not cover the structure here, but a little bit on the classification, because flagella could be used for classifying the organisms. So flagellum, as I mentioned, is a long thread, like here. This is all flagellum. And when it beats, when it moves, the bacteria moves with it. On the basis of this flagellum, thread-like structure, we can classify bacteria. If a bacterium does not have any flagella, we call it atrichous. Trichous hai, this means hair. So A means not. So there is no hair-like structure, there is no flagellum. Atrichous. Then, if a bacterium possesses many of these flagella, then we call it as a peritrichous. And these flagella are distributed around the cell. They're called peri, mean around. So peritrichous, distributed over the entire cell. On the basis of this flagella, we can call bacteria as polar also, because if they're on the ends, like the poles, we call them monotrichous. If there's one hair-like structure, we call monotrichous. It could be amphitrichous. That means amphi mean both, two. So if the structure is present on both sides, we call it amphitrichous. And this structure hair-like structure could be one, or could be a tuft, like bunch of them. So they are called lophotrichous. Could be on one side, could be on the other side as well. So in summary, the structures that are outside the cell wall include glycocalyx, which we called, if it is firmly attached to the cell, we call it a capsule. If it is not firmly attached, we call it a slime. And then the second is a hair-like structure, what we call flagella, used for movement by the bacterium. The same flagella, when it is present in the spirochetes, we call it axial filament. Again, it is used for motility or mobility of the organism from one place to the other. And then there are small hair-like structures all around the cell. We call that fimbriae. Fimbriae, we will study in a later lecture. They also are used for attachment of the bacterium to surfaces, much like slime or capsule. Hilli are special structures, not very many, sometimes only one, sometimes two. They could be present up to 10, or even, around 10. And their function is basically to transfer DNA from one organism to the other.